So let's take a look from the bottom up. What are the instructions themselves? And we're going to start, like I said, from the bottom up, from the nucleotides. And nucleotides are monomers of DNA strands, right? So they're the sort of letters. We'll be talking about this later as well when we talk about proteins. They're sort of the letters that make up the words that give the instructions. Those are called nucleotides. They're the individual monomers in a DNA strand. From there, 3 nucleotides in a row are called a codon. They're 3 base pair segments, so 3 nucleotides. And what they do is they instruct the addition of a single amino acid. So a codon, a 3 base pair sequence, says, make this amino acid. From there, a gene is a section of codons. It's a really long section of DNA, and it codes for a specific trait or a set of traits in the form of proteins. Codons string into genes, and genes give instructions for making specific proteins. And once you string together a bunch of genes, you've got the genome and you've got the chromosomes, right? Chromosomes, if you think about humans, right, the human genome is made up of 46 chromosomes, so 23 pairs. And the genome is the entire DNA sequence containing an entire complete set of inheritable traits. So you saw in your book, right, that if you took all of the DNA in a single genome, so all those chromosomes, and you just stretched it out and you laid it out end to end, you'd be able to go back and forth to the sun more than once. There is tons of information inside of a genome, right? So the idea is nucleotides make codons, lots of codons together make a gene, lots of genes together make chromosomes, and all of the chromosomes together make a genome. And those are the instructions. And the end result of this is proteins, right? The genome, in its chromosomes, in its genes, tells the cells to make specific proteins. And proteins are for a lot of different purposes. Proteins can speed up chemical reactions, in which case we call them enzymes. Proteins can have structural functions. So if you think about your body, it's mostly made up of liquid components, and yet you're not floppy and liquidy, right? You have proteins that are keeping your cells from being completely liquidy. Proteins also act as messengers and signalers. So insulin in your body actually signals your body to have a certain physiological response. Insulin is a messenger protein. Proteins also can get broken down to build new proteins, so those amino acids get recycled later to do new things. And proteins basically do everything your body needs to have done. So DNA is our genetic database, right? Nucleotides make codons, and codons are just little 3 segments of DNA. Codons make genes, and genes are long strings of DNA, and genes make chromosomes, and chromosomes make the genome, and the genome is the entire DNA that gives the entire instructions. So DNA is just a database of a way to make instructions. And the basic units of DNA are a phosphate, right? If you notice up here, PO4 3-, and if you think back to chapter 5 when we started talking about ionic compounds, that is the same phosphate ion. Phosphate is found in your DNA. It's part of a nucleotide. There's also sugar in DNA, and it's called deoxyribose. There is a sugar called ribose that just has an extra O here, so it's OH instead of H. Deoxyribose is the version that's in your DNA. DNA also has bases, and there are 4 different bases. There's adenine, guanine, cytosine, and thymine, and we go with those by their letters. So we call adenine A, guanine G, cytosine C, and thymine T. I'm not looking for you to recognize these chemical structures. I'm not going to show you nucleobases and say, is this one guanine or is this one thymine? That's not what we're interested in. But I do want you to notice that they have ring systems, right? There are always 2 rings next to each other if we're talking about adenine and guanine. And there's cytosine and thymine, which have 1 ring. Right? So there's a phosphate, there's a sugar, and there's a base that are part of DNA. And what happens is a single nucleotide is made of a phosphate group hooked to a deoxyribose, hooked to a base. That's a single nucleotide, phosphate, deoxyribose, base. And if we have a string of nucleotides hooked together, right? I see phosphate, deoxyribose, base, phosphate, deoxyribose, base, etc. So I see 1, 2, 3, 4 nucleotides in a string. Right? This is how DNA gets built. We're stringing together a bunch of nucleotides. And DNA, right, stands for deoxyribose, so it's got the sugars. Nucleic acids. Our nucleic acids are these guys. What about the structure of DNA? Once we get a whole big string of nucleotides, thymines, adenines, cytosines, guanines, they can come in any order, right? It's not TACG, TACG the whole way through. We'll get to that in a moment. But just a whole string of nucleotides together, there's a structure to it. X-ray diffraction done by a woman named Rosalind Franklin in the 40s or 50s discovered that if you looked at DNA in a certain way, this is like looking down the top of a DNA strand, and you see this kind of familiar X pattern. And her work was taken up by two guys that sound more familiar to you, named Watson and Crick. They all said, Franklin first and then Watson and Crick later, that this had to be what's called a double helix. It's like taking a ladder and twisting it. And Watson and Crick proposed that the distance between one base pair to the next, so one nucleotide to the next, is about 0.34 nanometers, 0.34 times 10 to the negative 9 meters. This is a pretty small distance, right? This is a nanomolecule, nanoscale. And they said that a complete turn of the ladder, so from one point to coming back to where it looks the same, is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 base pairs, right? And that is the distance in a complete turn. That takes 3.4 nanometers. I'm not looking for you to know the distances or memorize those, but I do want you to know, right? DNA is a double helix. It's a pretty short distance from one base pair to another. It's a longer distance from one base pair to when it makes a complete turn and comes back around. What about the nucleobases? You notice on this previous slide I see TA and CG, but I never see TG and I never see CA. How did we figure that out? Well, there's a guy named Chargaff, and your book refers to it as Chargaff's rule, that basically looked at the percent base compositions in DNA. So you could analyze a strand of DNA, even if you didn't know its exact structure or its exact sequence, and you could say, what percentage of that DNA was adenine A, what percentage was thymine, etc. And basically what Chargaff saw is no matter what, adenine and thymine and guanine and cytosine were present in the same amounts, right? It's about 31% adenine and 31% thymine, 19% guanine, 19% cytosine. Or when it changed, like if I look at a bacterium, 28.4 and 29, or 21.0 and 26, right? Adenines and thymines were present in the same proportions, guanines and cytosines were present in the same proportions. And what that means is that the adenines must always be pairing up with the thymines, and the guanines must always be pairing up with the cytosines. If it weren't that way, we wouldn't see this lockstep on proportions. And so that's what happens. And if you look at the structures of adenines and thymines, they hydrogen bond together really well. That's what these nice little red lines are. Those are hydrogen bonds between the adenine and the thymine. They fit, right? There's 2 hydrogen bonds between A and T. Now if I look at G and C, there's spaces for 3 hydrogen bonds. Adenines and thymines were made to fit together. They have 2 hydrogen bonds that lock them up. Guanines and cytosines have 3 bonds that lock them up. Now that doesn't always mean that DNA quite follows those rules, right? Sometimes there are problems that occur in a DNA sequence and you have what's called a mispair. So sometimes you do have a GT mispair. This is not the way they are supposed to line up. This is a problem, and it's actually a problem in the code. But mispairs can occur, and that's part of why you're seeing slight differences, right? 19.1 versus 18.4. But on the whole, the right way of pairing things is A to T and G to C. So if we had a DNA sequence, right, if it was TAG, then we must have ATC to match that up. Right? DNA is a double-stranded helix all the time, right? A double helix means that there are 2 strands, right? There's one strand and there's its complementary strand. But when DNA replicates itself, what it actually does is it turns one strand into 2 new daughter strands. Right? In this picture you see that at the bottom of the DNA it's partially unwound, it's unhooked itself. And so you have one strand hanging off and another strand hanging off. And what's happening is your body, your cell, is synthesizing the complementary strands. It's synthesizing the puzzle pieces that fit. And the process continues, and we keep unwinding this strand and synthesizing new ones until we have 2 complete separate daughter strands of DNA. This is what happens in your cells every time they divide, because each cell carries its whole genome with it.